So thank you for the introduction, Lale, and it's great to be here. This is my third time presenting at this symposium. I really like coming here, meeting colleagues, and meeting everyone here, so talking about what are the next steps and where this science is going, because I know everyone really wants to know what can we do next. So what I'm going to talk about in the next 40 minutes or so is give you some idea of why this initiative has been taken up by NDF and how it will help us in going in the direction of gene therapy. Because I think getting down to understanding the disease pathology and how it actually affects the entire sort of the physiological and the biological systems is absolutely critical as we keep discussing the gene therapy issue and what are the right strategies for gene therapy. So with that, am I doing this right? So I'm not going to talk a lot about this slide, but we have talked about, in the morning sessions, we have heard about what are the possible roles for the GNE across the different pathways and what we are actually looking at. What I wanted to just point out is that we are looking at 2 or 3 different roles here. One is the chemomechanical role, the other one being in the Golgi with the hypersilation, and then also in the nucleus. So the question really has been discussed again and again, and we see this in the literature with many, many publications, is that what is really these mutations, how do they cause the muscle pathology of weakness, wasting? It's really not clearly known. And how can we get to the bottom of this? As we start planning and looking into the different approaches for treatment, clinical trials, and then of course the big question of where do we address the treatment at what level? Do we go down to the real DNA level, the RNA level, or the actual protein level? So with that in mind, about 3 years ago, and I'm going to talk about some of the data I showed last year, and this is really important as I talk about why whole genome sequencing is important and why this initiative was discussed by the scientific committee here. About 3 years ago, we started this project at my previous laboratory, Emory Genetics Lab, talking about how can we identify these patients for clinical trials. As we have talked about it this morning, it's a rare disorder. So that means it is underdiagnosed. It is hard to find individuals to enroll in clinical trials many times. So with 2 main questions in mind for treatment and management and clinical trial recruitment, we started this LGMD panel. Now the first thing I want to point out to everyone here is how much science has advanced since the time we actually did this panel. In this panel, we had 36 genes. Today, if you go to the literature, there are about 80 plus genes which cause neuromuscular disorders. About 10 to 12 new genes are reported every month now, and it is really hard to keep track of what is going on. Many of you who have got this diagnosis were tested at a time when only single gene sequencing was done. And I'll point out to you why it is important now to go and do whole genome sequencing, and just doing single gene sequencing to identify and diagnose the disease is not enough anymore. So in this panel, as I mentioned, we recruited, it's actually 5,000 plus patients, and this paper has just got accepted for publication just 2 days back. So it will get published, and probably we can circulate it as well once it's published. This is a list of genes. These were the genes that were known at that time. We managed to include all the genes. You will see the GNE gene on there. There are many genes which are across the muscle pathology, which are included in this panel. And it was a next generation sequencing panel. It is a technology that is better than the sequencing technology that was available to us before this, when single gene sequencing was actually done. So what I'm going to jump into now is results from this 36 gene panel and how it actually helped us, guide us, and make that decision that whole genome sequencing is absolutely necessary at this point. So let's start looking into the findings itself. What I'm showing here is across the 36 genes, there are recessive disorders, so both parents being carriers and the individual inheriting 2 mutations from the parents. dominant disorders, X-linked, and then negative cases. What you see here is that we had a diagnostic yield of 27%. Most of these cases, when they were sent, the physician was not able to tell us, go for this gene, or I think this is this particular disorder. Extremely heterogeneous. We just did the sequencing, we found the mutation, we went back to the physicians, and they said, yes, this fits the clinical description. So that is point number one. The other thing I'm going to point out from this slide is the term there, variants of uncertain significance. This term was coined by the American College of Medical Genetics in the sequencing variant guidelines. What it essentially tells us, like people like me who look at the sequencing data today from all the available databases, in spite of everything that has been done, we still cannot decide whether this particular change causes disease or not. So we leave it as a variant of uncertain significance. So what does that mean? Because once we issue a report like that, I'm not helping anyone, right? The physician doesn't know how to tell this to the patient and what to do after that. So we'll get to this question, again, addressing through the whole genome sequencing project. So this was our sort of final result in the sense of what we found in terms of the data here. And you can see that we had GNE, 2% of the patients had GNE myopathy. Now these are definite pathogenic changes or mutations causing disease. I'm not showing you here the variants of unknown significance. And in the next few slides, I'm going to talk about how this data actually now keeps changing as we dig more and more into it and start looking at the gene level. You see dysperlin and calpain-3, that was an expected result. ANO5 really threw us off as well. This is a fairly newly described gene, but we did find a lot of patients with ANO5 mutations as well. So let's look at some of the other genes and what our data showed us. We found a lot of homozygous mutations for dysperlin and FKRP. There are some common mutations, so that was not a big surprise. But what we did find was that the GNE mutations, we did not see a lot of homozygosity there. There are some common mutations in the GNE gene, which we detected, like what was presented this morning. The Indian mutation is obviously one of those mutations. But one of the interesting things which I want to talk about is that though we identified a lot of unique pathogenic mutations across the spectrum in the different genes which we tested, and you can see here number of patients identified per gene. You can see that it starts with dysperlin, calpain-3, and then starts tapering off there. We started digging into what were the actual mutations that we found, where they were truly pathogenic or disease-causing or not, and then how many variants of uncertain significance which we found in these patients. And this data is absolutely staggering because there is so much more that needs to be done before we actually start doing a real gene therapy clinical trial. So let's look at that. Let's now just focus into GNE only. So what I'm showing you here is all the different mutations which we found in the GNE gene. Now, you see two terminologies here, and don't want to throw you all off. Pathogenic is a true mutation, and likely pathogenic essentially just means following the guidelines given by the American College of Medical Genetics, where the data is not fully there, where we can move it to pathogenic. But it is, just for the sake of this discussion, it is pathogenic. And I like that Wayne's nodding his head. So you can see the mutations which we found there, and what the sort of big question is, these are the mutations which we saw in the patients which we were able to with certainty say have GNE myopathy, give the clinical results back. But then what about these diagnostic hurdles? Think about those patients where we had to issue reports with variants of uncertain significance, and then the patients who did not, in whom we did not find anything, the negative patients. So how do we start addressing this? So let's look at the variants of uncertain significance. What I'm showing you here is two pie charts. First one is just telling you across, not just GNE myopathy, neuromuscular disorders, across the entire spectrum of inherited genetic diseases, how many variants can we actually, or changes in the DNA, we can actually classify. And you can see it is close to 50%, Today, we just simply cannot tell in a clinical lab after doing testing whether the DNA change actually is causing disease or not. Now, the same taken now and extrapolated to the limb girdle cases, which we did. And you can see it's somewhat similar, a little more variants of unknown significance, but it's not terribly different. Now, let's look at GNE, and it looks the same. So we had about 25% pathogenic changes, likely pathogenic, and a huge chunk of variants of uncertain significance, which we do not know what they mean, do they actually cause disease or not. These are now remember, these are not healthy individuals we tested. We tested individuals who have some sort of a clinical presentation, and that's why they were tested expecting to find a mutation in some gene. So these kind of DNA changes obviously need to be studied, addressed, so that we can give a proper diagnosis and issue a clinical report. So that is problem number one, finding answer to variants of uncertain significance, and what do we need to do as a next step to sort of confirm the diagnosis so that these individuals can access clinical trials or gene therapy trials when they are available. Now let's look at the next problem. So as at the start of my presentation, I said that most of you must have had single gene testing. Your doctor must have gone gene by gene by gene. At that time, the technology was not there where we can test a number of genes at the same time. So a single gene was tested, it was negative, then another gene and another gene was tested. So single gene testing was a norm at that time. When technology changed, we are now able to test a whole bunch of genes at the same time, not just a whole bunch of genes, our entire genome, and we'll talk about that. So what we found in this sort of cohort is that not only we found 2 mutations in the same gene in the case of GNE myopathy, for example, but we also found mutations in other genes. And this is where the story starts changing, and it becomes interesting and important from the patient perspective of why we need to look at other genes when we have a diagnosis for a single gene, and that has been known for a very, very long time. So essentially what you see here again is this pie chart which shows variant contributions, and we call it multigenic combinations or contribution. And I have shown you like different types of combinations, pathogenic or known mutation versus mutation, mutation versus variant of uncertain significance that needs to be further resolved. Now, let's look at that a little bit further of exactly what am I trying to say here. Okay, so we know that when we do GNE myopathy testing, we are looking for 2 mutations. In this case, what you see on one side, no phenotype, is when the one mutation is there, and that individual is a carrier. When the balance tips in the other direction, where you have 2 mutations, both little a, in this case, little a is a disease-causing mutation. What I'm saying is from our data, we were able to find that not just little a, but we also found mutations in other genes. You see that little b with a star and a little c with the star. I don't know what that means. Does that actually modify the clinical presentation? Does it make some individuals have a rapid progression? Some have regression. We have all sorts of examples. I'm going to show you some actually GNE myopathy patients' data where we have all these different examples where 2 mutations in the GNE gene and another one in a different gene. So looking at the multigenic inheritance just across the board, not just, I'm showing you first data on just all across the genes which we detected. And you can see ANO5, GA is Pompe disease, we have calpain, we have got sarcoglycans, we just see it all over the place. So this is about 25% of the patients which we tested. Now remember, I did only 36 genes, so there could be many more other mutations I don't know about. But we were able to show that this is real. Now, what is the next step? Because this is a big paradigm shift in our field. So the way to look at sort of inherited genetic diseases or just genetic diseases in general is you have the monogenic, a single gene, 2 mutations, 1 mutation causing disease. And then we have the chronic or the diabetes and the asthma, which are thought of as diseases or disorders called by a general imbalance across the genome by various contributing genetic factors. Somewhere in the middle, I put this multigenic group. which we have to prove that this is a real mechanism. How do we prove this? The best way to do it is take a muscle biopsy, do RNA sequencing, do gene expression studies, and show that these various mutations in different genes are actually sort of influencing the pathology of the disease itself. And I have a grant from MDA right now where we are looking into this phenomenon to actually prove it. So now let's look at the GNE cases itself. Where did we find multigenic evidence for GNE? And I don't know if you can see this clearly, but I've given you about 7 examples there with GNE, either a pathogenic or a variant of uncertain significance. Now, when I put a variant of uncertain significance, sometimes when we are doing analysis, we know that this is very likely to be a mutation. But we simply cannot put it on paper because we don't have all the biological evidence collected with us to prove it. But you can see that what I put there on this slide, so out of the 64, I selected 7 because I wanted to show you the range of other genes that could be involved. So we have desmin, we have calpain, we have LMNA, Titin, POMT1. I mean, it's just really across the board, we found mutations in different genes. So now we talked about variants of unknown significance. More than 50% of variants we could not classify. Number 2 problem is this multigenic inheritance, but that too from a very tiny panel of 33 genes. What if I had looked at 80 genes or across the genome? Can I now start digging into the data and say that why is this differential expressivity there? Why do some patients have a faster or rapid progression, some do not with the same mutation? And this is sort of published in the literature. So digging even more into the GNE data now, what I've shown you here is a number of confirmed GNE cases and all these various combinations that we saw. So we had real sort of confirmed cases, 8, but then we have all this variety of different combinations which we have found. What we have done as a next step is written to the physicians who sent us these patients so that we can get blood samples, we can get the muscle biopsies, and actually do RNA sequencing to show if this is something that is real or not and how this is actually affecting the disease pathology.
Can I ask a quick question just on that top line of that last slide?
Yeah.
When you say confirmed GNE, that means 2 pathogenic mutations
Correct.
identified. Okay.
Correct.
And nothing else.
Yeah. I mean, the rest of them, I can tell you that they are very likely pathogenic, but I don't have all the evidence to say that. But 8, 100% reported in the literature as well. So now, obviously, the next question that is coming to your mind is, can you tell us about the clinical presentation of some of the patients who possibly had this multigenic inheritance? So just because of the size of the slide, I did not put all the 7. I've just put 5 here so that you can actually read what I've written, hopefully. You can see the clinical presentation and progression in this column here, which sort of shows you, the kind of the subtle differences. Even if the mutation is the same, you start seeing this kind of difference in pathology and the actual progression of the disease itself. We have everything from unusual myopathy, slow progression, some had cardiac issues, may not be just associated with any of these genes. It could be something completely different. Weakness, and this is just spread so widely across all the patients we tested that it really sort of got us thinking that doing single gene testing, identifying the 2 mutations, and then jumping into clinical trials really may not be the right approach, because we know that if the trial fails, we are starting to look for all the reasons. So why not prepare ourselves well before jumping into a clinical trial so that we know who is going to respond, who did not respond, and why did that happen? At least to the extent of the knowledge we have today, let's get all this data together. So with the NDF scientific committee, we started talking about this. Can we take this next step while we are actively researching into and planning gene therapy as well? So I'm going to show you the amount of work that goes into every single case that in my research lab and also in the clinical lab at PerkinElmer of what we are trying to do when a single case comes to the lab. And this, I deliberately put this case up because it has been discussed in the last 2 meetings as well. This is a very unique kind of a case. because it shows the entire mutation spectrum of the gene. It's a common Indian mutation as well, and I think it has been published, this particular case. So you see the common Indian mutation there. We did not find a second mutation. It actually turned out to be a big deletion. A chunk of DNA was gone. We had to run a different kind of a assay to detect that deletion at that time. It was a microarray. You can see that little red bar there, which is just showing you the deletion that happened. So then we started looking at what these two particular mutations are actually doing. And I think Wayne talked about that on the panel, that every disease, every gene, every mutation acts differently on that protein or the enzyme. And this is a very good example. So the common Indian mutation actually has a different effect than the deletion itself. So in this case, the deletion actually results in a monoallelic expression. By that I mean that one of the allele is gone and one is expressed, which is the common Indian mutation here. Then we did a gene expression array to actually prove it that the gene expression had reduced. And you can see that here with control versus GNE, where the actual expression of GNE had reduced. So that was one way of showing that the deletion actually did something. What about the missense change? And this is where it starts getting tricky, because you can actually spend an entire year on just taking two mutations, and this is my postdoc's work for about 6 months, looking and digging into what the mutation is doing to the gene and how the expression is actually affected. So as I said, the deletion actually sort of results in loss of one copy of the gene. The missense change has a very different effect. You actually don't lose one copy of the gene. In this case, what happens is that it is situated right in this crystal structure which you see. It actually affects the kinase activity. But it's not completely abolishing the expression of the gene completely. So this paper is also just accepted for publication. But what I wanted to show you all is that every gene, every mutation has its own characteristics. Every gene has got a different content, and there's a lot of work that has to go in after identification of the two mutations in the gene to put it in a database so that it's available when we actually go to a clinical trial. So let's jump into whole genome sequencing. So just to summarize, we are addressing several issues: variants of uncertain significance, contribution of more than one gene by knowing the primary gene. In this case, we are talking of GNE as a primary gene. Are there other genes that contribute to the progression of the disease? And number 3 is that every mutation has its own characteristic effect on the gene expression itself. So that is kind of the foundation of why we decided that whole genome sequencing is really critical and important as we plan gene therapy. So let's look at that a little bit. In just the whole world of genetic testing as such, the paradigm there's a huge shift now. Very rarely physicians now order single gene testing. It's either a gene panel, exome or the genome, and I'll talk about what exome and genome means. It is sort of going to this higher level of testing, and we'll discuss that. This is really important because most of the clinical disorders nowadays we know are heterogeneous. What do I mean by that? There are overlapping clinical presentations. There's rapid progression, slow progression. Some individuals are asymptomatic. We know of such individuals as well. So single gene testing probably is not the right approach. It is still done in certain cases like cystic fibrosis, Duchenne muscular dystrophy. The clinical presentation is so strong that they don't need to do this kind of expanded genetic testing. So let's look at it a little bit more of what we are trying to do when we are trying to go away from single gene testing to gene panel, an exome, or a genome. Really trying to create a structured framework and actually getting all that data together in one single database at the phenotype level or the clinical presentation, the disease level, the understanding the pathology, the gene level, which Wayne already talked about, and then the different pathways that are involved in this process. And can we bring this all together as we start planning the treatment and the trials? So this is from one of my publications, and because I work on neuromuscular disorders, we sort of started putting this database together in my research laboratory. So let's look at the genome now. I think I've sort of hopefully created a foundation of why we are doing this. So genome is all of our DNA, right? It contains genes, which is, we call that the exome, 22,000 genes in our body. But look at the percentage. Only about 1 to 2% of the entire genome is this 22,000 genes. The rest of the genome, we don't know what it means today. But the technology has gone ahead of us. We can actually sequence the genome at the same cost as, probably sometimes cheaper than doing exome nowadays. So obviously the move is, I want to sequence the genome. Many individuals come to us. In my lab, we do both clinical testing and healthy genomes, individuals who are healthy but just want to know, what could be coming. So what we do essentially is look at the 22,000 genes. But now take it down to another level. Of the 22,000 genes, today we only know about 5,000 to 6,000 genes, what disorders or diseases they cause. So there is so much work that remains to be done to understand what these genes are doing. Take it down another level, out of those 5,000, only 2,000 genes actually are truly associated with pediatric disease, and the rest range from pediatric to adolescent to adult diseases. And then, what sort of many of you got tested for is that one single gene from that 5,000 genes, and in this case, GNE. So hopefully I've laid it out to you that how much is there to do, but the technology is there today. We can sequence the genome. I've sort of put forward the questions we are trying to address by doing genome sequencing, and how we are, now I'm going to show you what that plan is and where we are going with it. So as I mentioned, the technology is there to generate the data. Today, we can take an individual's DNA, we just shred it to pieces, put something called adapters. They help us guide that sequence, the DNA through the system, and we can generate data for the entire genome. The next step, obviously, is to take that data, the sequence of the genome, and try to now start making sense out of that data. So basically what I'm showing you here is generating the information and then taking that next step, which is analysis and interpretation. Now there are two ways to do it, right? When we get a sample, we actually get the clinical information, and we put that clinical information, we call it gene prioritization. So if it is a neuromuscular condition, all the neuromuscular genes should get pulled out. It's a software which just sucks out all the genes for us to look at. So that's step number one, and then you start drilling from there. Now, as this process is going on, it is a very iterative and interactive process with the physician. If it's a sample coming from Wayne to me, I would call Wayne and say, hey, I see these 2 mutations in these 2 genes, but then I see this other mutation too. What do you think? And it's a very interactive process of coming to the answer. You might have read in general media where babies are now getting sequenced, babies in NICU, to come to a faster diagnosis, and that's where the genome sequencing is very useful, because you're not going gene by gene, and you can do this process in 7 days now. This is really phenomenal, because it's when our genome was sequenced 18 years ago, it was several $1 million down to $1,000 now. So it's a huge difference. Now this is just sequencing. Interpretation is a skill set that is a whole added cost to what we are doing. So this is where we came to trying to answer these questions and how we can get this together for NDF and address the question of GNE myopathy, as Lale and her team are planning the gene therapy projects. So if you just look at the bottom of this slide, essentially I've already told you there are 12 new genes that are identified every month. It is a very difficult thing for individuals like me who run labs to even keep up with this. But essentially what we are trying to do here is, first of all, shorten the time to diagnosis. I think it's absolutely critical. No one should have to wait for years together to find out what is exactly going on. But then identify these disease modifiers, biomarkers, and share it with the wider community. NIH, there's so much research going on. Can we bring this all together and start sharing it so that we can actually sort of design some proper clinical trials? So this is where we have ended up right now. It's funded by NDF and partially funded by PerkinElmer as well. I run the global labs for PerkinElmer. The advantage of running global labs is we have a lab in India, 2 in China, 1 in Malaysia, and 2 in US. What happens is when sequencing is done, all that data gets pulled up into a cloud, and we can actually see frequencies. So what is common in the Indian population may be rare in the Caucasian population. Interpretation gets easier. The Chinese population, because China does not allow DNA to come out, sometimes it's very hard to do interpretation of an individual who's living in the US, is Chinese, and I don't know if that variant actually causes disease or not. So we are generating this database at a very high level so that we can come to these answers faster. So whole genome sequencing, metabolic profiling, and then compiling all this data into one single database and make it available for future trials. This database is absolutely critical. The design of this database is important. Full clinical information, no information is trivial. I cannot say this enough because when we get samples for testing, sometimes physicians tend to leave out information thinking this or that is just not important. Everything is important when we do data analysis. So as I said, the objectives being if the mutations are known, we'll confirm the mutations, confirm that there are no other contributions from other genes, or if there are, document that, start understanding what is this contribution, how it will affect the disease process, and then how the individual will actually respond to different therapies as and when they are being designed and become available. So the goal being, this wide goal, but I think it's important to sort of bring it down and start sort of acting on it. And here's where the family members become really important because they are either carriers or there is a wide range of clinical presentation or progression within the same families. And such families are absolutely critical for this kind of research to document it so that we can actually try and understand what exactly is going on. Then we have sort of taken it down to another level. And this is not really sort of separating any mutations out or anything like that. We are starting to look at everything. Can we just take the 3 to 5 most common mutations and start with that? How do we sort of collect family members? Because there's a lot of education that needs to happen around it. We need to do some sort of justification of why it's important to contribute to this research. And then also start looking at, we heard about the MANAC therapy and all these different things that are going on, document that in the database. Did someone respond? Did someone not respond? Well, the individual did not respond because that other mutation was there in this other gene. Can we take the muscle biopsy and do RNA sequencing and see what the gene expression profile is? So there's so much we can do with this whole genome sequencing project. So we have started the project, been talking a lot to the scientific committee. Moncol is helping tremendously, sort of helping me also sort of guide through this entire process. We are starting to look at a variety of mutations. What is the right way to do this? What are the controls? How do you do that comparison? Because obviously we want to draw the right conclusions from this project. So this is where we are. I think this is really important. I know most of you are sort of really want to know about clinical trials, gene therapy, but it is absolutely critical that the foundation is solid because we want to do this right, and that's why this project is really important. So just in summary, I've shown you the data from the gene panel, what pathogenic or mutations mean, what a variant of uncertain significance means, why is it important to study it. And then we have initiated the WGS GNE Myopathy study, really the NDF initiative, and I'm really glad that NDF has decided to do this. So with that, I'll stop and see if you guys have questions. Thank you.
Where are you, what stage are you in collection of the data for the GNE Myopathy?
So we have started collecting the patients. We have about 30 right now. Sequencing will start soon. Now, like I said, it takes 7 to 10 days to generate data. The next step is to do interpretation analysis, collect all the clinical information, previous testing information. So all that is in the process right now. Obviously, we have not recruited the full sort of number of patients that are needed. We've just started with that.
And so you need more patients, right?
Absolutely.
So all of us in the room can participate.
Yes.
Thank you. Does anyone have any other questions for Dr. Hegde? One over here. One moment.
Hi, I have one from Facebook Live right now. How do we go about getting this testing from Jeff Johnson?
Lali, do you want to answer that?
How are we going to do the testing for,
How to participate?
how can he participate? Well, a couple of ways. One, today we're collecting samples from the people who are here who have volunteered. And then I know that Perk and Elmar can also send you a kit
Yes.
at home. So please send an email to info@curehibm.org, and we will put you in touch with the right folks over at Perk and Elmar.
So I did not mention this, but we can do this on saliva. We can do it on blood. We can also do it on a dried blood spot card.
Okay,
So
great.
finger prick.
Yes, we have another question over here from Dr. Bhattacharya. One second.
So you mentioned about metabolic profiling. Are you also going to do metabolomics of these samples?
Yes, so that has been planned, not as a first step. I think we'll go through the whole genome sequencing, we'll analyze all the data, and we will do metabolomics. I'm actually putting together a metabolomics platform in my lab, and which could easily be extended. So we will do it.
For the sake of a lot of people who then come to me later and say, I didn't understand what that meant, can you explain what the difference is between metabolomics and the other sequencing? Thank you.
So what we are trying to do at the DNA level is just see what other genes could be contributing. What we want to see is the products of those genes. Are there any biomarkers we can go for? So there are two ways to look at it. Is just to look at the metabolic profile of different markers, but also see if there is some marker which we can use for future tracking of success of clinical trials or not. And technology is available there today. We can use mass spec. It's one terminology. The other one is NMR spectroscopy. These are very sensitive methods to look at for these biomarkers.
A very important project, and this is really due to occur now. My question is, are you planning to look at the epigenome? And if so, you may want to try to get blood, to get DNA samples from the same source,
Yes.
not saliva, blood, different sources. Because epigenetics may get very important for GNE myopathy, because it is already in the literature that the GNE gene expression is actually regulated by methylation. So that would be one of the first profiling I would do if I had GNE myopathy DNA available, and see if there is a genotype-phenotype correlation.
Yeah. Absolutely agree with you. Obviously, the ideal sample is a blood sample, to take the blood sample, and we can sort of store the blood sample appropriately as well, so that these future studies can be done. And even then when I'm doing sequencing, I can do a methylation sort of profiling at that point too. It really depends. Some individuals prefer giving saliva, if they're not in the country, or sort of different things. But I think one of the things which is very important is this database, right? Because if I get this all together properly into a database, and as you said, as we learn more and more, we can approach the individuals again as well. But absolutely agree. I think blood sample is the right sample to do.